Sorry. Okay. Yeah, so you'll want to watch the recordings and then take the quiz every week. So there's a lot of things in here that I'm really not going to talk about because it's not really, it doesn't really pertain to the, the Zoom presentation. Um, so really, there's drafts don't pertain because we don't do any drafts of anything. You just get quizzes. Extra credit, there's no extra credit opportunities available for individual students. If I do give any extra credit, it'll be available for the entire class, and the entire class will be informed. Um, yeah, so we will talk about attire because you should be aware of what attire should be like for the laboratory. But needless to say, because we're not in the lab, I'm not going to worry about what you wear, obviously. Um, yeah, yeah, so I think a lot of this we won't really cover. If you do need accommodations, please make sure you do contact Student Support Services. That's Hillary Gregory, and there's the contact information. Um, Statement regarding academic honesty. Yes, please take all the quizzes from your memory and understanding of the material. So that is appreciated. Yes, with the quizzes, again, take everything from your memory and understanding. Um, yes, you can, basically, we will have the quizzes graded immediately, so you should have them as soon as you finish the quiz, you should also receive the grade. Okay, any questions about that? Okay, so this lab is given not in Lion Center 204. It will be instead synchronously online on Tuesdays 3 to 5 p.m. and Wednesdays 3 to 5 p.m. This is the schedule, and the way that you're going to be graded is by lab quizzes. There will be 10 quizzes, one for each one of the scheduled labs. And as you can see, we don't actually have labs next week because Wednesday does not meet. So the Tuesday lab won't meet either. But then all of the other dates, we will have labs. Um, so please make sure you watch the video, or please make sure you come to this lab synchronously, and then please take the quiz before Fridays of every week. Okay. Um, so if a student takes 10 of the quizzes, the average will be determined after the lowest quiz score is dropped. So generally, the reason we don't do makeups in this lab because all of this material is live. So if a quiz is missed because you were sick or because you were absent, that you were physically absent, you couldn't get to the actual laboratory materials on Moodle while it was available. Yeah, we'll have to talk about doing some sort of makeup. So please try to complete everything by Friday night. Okay, that's the best thing, and I think it's plenty of time. You're in the Tuesday lab. That's a lot of time from Tuesday until Friday. Tomorrow, Wednesday, they will have the same rule, but they have a little less time. Okay. Any questions about that? Okay. So if you have any questions about the lab, please let me know about how it's basically functionally going to be actually carried out. All right. So we're going to take a look at. We're going to go on to start to look at the lab itself. Okay, so this is the first laboratory. And so some things you're probably already aware of. And as I said, even though we're not going to be in the lab, I do think it's important for you to understand lab safety. So everything you hear or see in this introductory lab, please remember it's fair game for the quiz. So lab safety is no food and drink in the laboratory. So we don't allow any eating or drinking in the lab. It's also important when you're in the lab not to actually throw food or drink um, containers away in the lab trash containers. If you do have any food or drink containers that you actually had the food or drink outside the lab, you actually want to make sure you throw it away outside the lab. The reason why is because there is an organization, and maybe you've heard about this organization. It's called OSHA, and it's a safety organization that is meant to oversee, laboratory safety. And one of the things that you don't want to do is show that possibly, anybody has been neglecting regulations. So one of the things is that they say is that they don't allow any food and drink in the laboratories. And so if they see things that are like, you know, uh, drink containers thrown away, if you have a coffee and, you throw away the drink container in the lab, it looks like somebody was drinking in the lab. Or if somebody, throws away, let's say, I don't know, a pizza box in the laboratory, they ate it outside the lab, but came in and threw the weight of the box. That would also look like somebody was eating pizza in the lab. So, you can't do anything that even makes us look like we could be guilty of violating regulations. So, so labs, they, even when they're not being used as a lab, so sometimes our labs are actually used for lectures only, they still have to be treated as laboratories because they are officially labs. And an organization such as OSHA could say, if they saw people eating or drinking in a lab, even though it wasn't being used for that particular course as a lab, they can actually basically charge the college very severe fines and even potentially shut them down. So it is a very severe problem. And usually OSHA, they do sort of do spot checks. So they just come into labs without letting people know that they're coming, just to make sure people are abiding by regulations. So clothing is really important for the laboratory. Um, Generally, clothing worn in the lab must be practical. No bulky clothing and sleeves. In the lab that we normally do in microbiology, we usually have these wonderful little things that I'm sure you've used at one point in time, and these are little Bunsen burners that have this nice little flame, um, and the problem is that it's very easy if you have a bulky sleeve or if you have some extra jewelry or something like that, it might get hooked on it and all of a sudden, oops, it falls over and then we've got this flame going toward people or maybe burning up some alcohol that might be there. So there is a lot of reasons why, there's definitely lab safety regulations that should be followed. Clothes must cover the knees, so you need to either have pants that cover the knees or a skirt that goes um, to cover the knees. It could be a laboratory coat. That would be fine too. Closed-toed shoes must be worn, which means no sandals or flip-flops, because things can fall on your feet. One time oh my goodness, we used to have these um, monocular microscopes. They were very archaic. That was a long time ago. And one time a student got something hooked into the cord and the microscope fell on her foot. She had a sandal on. Don't ask me how come. And so the actual girl had to go to the hospital because she did break a toe. Um, Miraculously, the microscope was just fine. It had nothing wrong with it. Hair should be tied back primarily because you don't want long hair, going into the flame. Hair burns beautifully, so you definitely don't want that to happen. Um, and you also don't want it to get into the stains either, or into any bacterial cultures. That all of that is bad. So lab coats, they are optional. If you were to be in there, you would have to supply your own lab coats. Sometimes I don't like lab coats because they are bulky. So I often, recommend that students are better off wearing, practical clothing, often clothing that they don't worry about if they get, stained on or something like that. Lab gloves are also optional. There's so many people that like different kinds of gloves. Some people are allergic to latex, so they use nitrile. Some people like powder in them. Some people don't like powder. Some people like a large one. Some people like a medium. So it's just easier for students to bring their own gloves. Goggles are also optional, but also goggles are another level of safety. So it is a good idea for students to wear goggles or some type of protective eye cover. Generally, we do recommend that students not wear contacts in the laboratory. If students typically wear contacts, they might want to switch to wearing glasses during the labs. Just because we have had experience where if a student does get something into their eye, or if they get something that's a volatile, sometimes there might be some volatiles. Sometimes the volatiles, these are things that get into the air that might be like chloroform. They're very volatile, and they somehow get between the eye and the contact lens. And when they do that, they can actually burn the surface of the eye. So it's a very serious situation. So sometimes wearing contact lenses when you're doing those sort of things is not a good idea. It's not even a good idea if you are wearing goggles, because those volatiles still get into the space underneath your goggles and between your eye and the contact lens. So the lab benches are, they're in our labs are pretty good. I mean, there's a lot of space, which is really quite nice. But by the same token, you don't want the space taken up by your personal um, uh, accoutrements, whether it might be a backpack or a purse or something like that. It's a good idea for you to be always putting all of those things into a locker outside of the lab. So anything extra like a coat or a backpack, anything like that should actually be put so it's not in the lab. It also makes it safer for people to move around in the lab. It also makes it less worrisome. If somebody were to spill a bottle of stain, hopefully then they wouldn't get the stain all over your backpack or something like that. Um, so the lab benches are typically cleaned very regularly, but still sometimes there can be microbes, chemicals, stains, other substances that can actually contaminate or damage your clothing and belongings. So you want to make sure that you clean up the lab bench if you spill anything, and remember that there can always be contamination on the lab bench, even if even if it really looks clean. If you do have any kind of spill or contamination, you can ask the lab instructors for assistance to clean them up. Okay, so it is really critical for each lab to follow all the lab instructions very carefully, because failure to do so could result in injury or exposure to contamination. And you should always ask if you're unsure of a procedure. So normally we have a lab safety agreement. We don't have to do that. Okay, so we're going to just introduce the light microscope. Obviously, we think that everybody is pretty familiar with using the light microscope because you had to do so for anatomy and physiology. But I think it's important to just make sure that students are reacquainted with it, because especially in this class, I mean, you won't be doing this um, physically, but it is really hard to actually find these bacteria. They're tiny. And if you don't have good laboratory technique, and if you don't have good technique with the microscope, it's very likely you'll struggle finding the bacteria. So it is a good idea to be very familiar with the functioning of the light microscope. So I sort of always like to start at the bottom when I'm looking at the microscope. I think because that's where the source of light comes from. Oops, that's where the source of light comes from. So I think that that's a great place to start. So pretty much there's a on-off switch right there. This is the source of illumination. There's just a regular incandescent light bulb in there, and that will actually shine light directly upward. The next thing that happens is that you get to something that is called the condenser. So that is actually a condenser lens. And for a lot of reasons, this is a very important part of the light microscope because on the condenser lens is usually a lever. And this lever on the condenser lens is called, it controls something called the iris diaphragm. Now, the iris diaphragm can either be all the way open or it can be closed down. So it's not as if it's completely closed. It's just much smaller in diameter. So when it's open, um, a lot of light goes through. When it's closed, minimal light goes through. Now, what's important about this, and you might have used it in anatomy and physiology lab, maybe you didn't, but when it's open, the light is more diffuse, and as a result, it decreases contrast. So it decreases contrast. On the other hand, if you have the actual iris diaphragm closed, it decreases the light coming through. And then as a result, it increases contrast. And in fact, that often is the situation that you're going to want to use when you look at the bacteria. You want to sort of close down the iris diaphragm so that you can increase contrast. This is usually the preferred situation to look at bacteria. It just gives you that much better of a viewing of the bacteria. Okay, so that's the first lens that we run into is the condenser lens. And really its whole purpose is to focus the light on the specimen. All right. The next thing that happens is you get to this mechanical stage. And it's very nice because the mechanical stage has these little clips on it that enables you to lock the slide into position. And you can actually, there are these stage controlling knobs on the side that enable you to move the slide left and right, back and forth. And you can actually see that there's gradations. I mean, if you wanted to, go back to a particular area on the slide at a later time, you could do that because you could just put the coordinates down, on your laboratory notebook and say, oh, there was something I wanted to take a look at or have your instructor look at a later time. So it's really good that, instead of having just to move the slide around with your fingers. All right, the next thing that we really come into is something called the nose piece, which is sort of a funny name, this nose piece. What it is it's a circular mechanical structure that the objective lenses are actually hang off of. So these objective lenses are the second lenses that you run into, and there's usually between 3 and 4 lenses that are attached to the nose piece. Usually there's one that is maybe a 10x, and that's called a low scanning. So this has to do with magnification, 10 times magnification. Sorry, you know what? That's not right. So there's usually a 4x, and that is a low scanning. Oops, I'm having all types of problems here. Low scanning. Then there's a 10x, and then there's usually a 40x, and then there's usually a 100x, which is usually an oil immersion lens, which means you shouldn't be using that one without using immersion oil. So that's typically, our 4 lenses. So there's a low scanning, 10x, 40x, and 100x. Okay, and their purpose is mainly for magnification. So that's really what they're doing. They're taking the light that has come through from the condenser lens, where the condenser lens focuses the light on the specimen. The specimen interacts with the light and usually causes the light to be diffracted, sent in all different types of directions. Then the light is essentially collected by the objective lens, and then the light is going to go up and then through these lenses, which are the ocular lenses, and they are the eyepieces. They actually do have a magnification as well, which is 10x. And so when you're actually viewing using the 40x, so if you're using a 40x lens, you are actually 40 times 10, which would be, 100. So 40 times, what is going on with this? Sorry. 40 times 10, which is going to be 400. So the magnification you're using when you're using the 40x lens, because of the ocular lenses, is actually 400. If you're using the 10x, it's 10x times 10x, which is 100. 4x times 10, which is 40, and 100 times 10, which is the maximum you can use for a light microscope, which is 1,000 times magnification. So the other things that are really important is you can adjust the distance between the lenses. So you can adjust the distance between the lenses on the oculars, which is really important because some people have eyes that are more wide set apart. Some people have eyes that are more closely in position. Then another thing that you have to understand is about the coarse and fine adjustment. Okay. Any questions on the microscope? Great. All right, so pretty much we did all of these. Okay, and the mechanical stage and the objective lenses. Okay, some of the most important things when you are using the lenses is, making sure that the lenses are clicked into place. I mean, a lot of times students will, be showing me something and things are all out of kilter because the lens didn't actually click into place. When you do put a specimen on the microscope, so when you have, the microscope and, you have, here's your objective lens and here's your slide, what you want to do is you use the lowest scanning lens first. And if you do this, you can bring the actual specimen as high up as possible. So you want to bring the specimen as high up as possible, as close to the lens as possible. And then you actually start to focus by actually focusing down. And that way there's only one direction to go. A lot of times you can get lost if you just put the slide on there. As you sit there and you say, oh, should I go up or should I go down? If you put it all the way as far up as it can go and it's not in focus, then, just by focusing so it goes down until you can actually see it, makes it a lot simpler because you don't have to question, oh, should I go up or down? Um, so you want to center your specimen in the field while viewing through the oculars. That's really important, especially as you're going to go to the next highest power. So once the specimen is centered and focused, you want to rotate the lens or the nose piece to the next highest power. So, when you're looking down through the microscope, you don't want your specimen here. So you don't want your specimen out on the corner of view. You want to bring your specimen so that it is centered. The specimen should be centered always in your view. And then when you go to the next higher magnification, if it's even ever so slightly off, so now you go up in power, you might find that your specimen now is over here. So you're going to have to, recenter the specimen before you go to the next highest power. So this is important because as magnification is increased, the field of view is decreased. So that's a really important concept to remember. As your magnification is increased, the field of view is actually decreased. So if the specimen is not centered, it might go out of the field when you increase magnification. So for example, if you have a specimen that's over here, when you go up in power, that field of view actually gets reduced like that. So now when you look at it, you can see that your specimen is no longer in the field of view. So that's why it's really important that you always center your specimen before you increase magnification. Okay, so this is pretty much the situation. This is just somebody diagramming it better than I do. With low magnification, you have this sort of field all the way out here. With the low magnification, you're all the way out here. But then as you increase your magnification, you're actually going to, come in here. So anything to the outside of that, you're not going to see. Any questions about that? Okay. The microscopes that we use in the lab are binocular, so it is possible to adjust those distance between your eyes. It's also possible to adjust the focus. So if you're a person that does wear glasses, but you don't want to wear glasses when you use the microscope, what you can do is you can actually adjust them so that they're adjusted to your eyes, because one of one of the oculars actually has a focus ring right on the ocular. So what you do is you use the one ocular to actually focus the microscope, and then you use the other ocular that has the focusing ring on it to focus the image that you see to your eyes. So I generally don't recommend students to do that. I generally recommend students to always use your glasses while you're viewing through the microscope, because what happens is then when you go look, like say if you were writing something down on a notebook, or if you were drawing something in a notebook, you would find that, oh shoot, I have to put my glasses back on, because even though I could see fine through the microscope, I can't see my notebook. So generally I recommend students to just keep their glasses on. Okay, any questions about that? Okay, another thing is a term that's called resolution. So resolution is a little bit different than magnification. So magnification is just increasing things in size. So in other words, if something is little. If you, you know, magnify it, it's, bigger. Okay. Resolution is something different than that. And a lot of times it's very important to not just have magnification, but to have resolution as well. So it's important to have both magnification and resolution. So what magnification is just that ability to make something bigger. But resolution is the ability to see the detail that something has. And the best way to explain it is sometimes, there might be 2 points that are very close together that, in fact, you might actually only see as what appears to be one point. But with resolution, you can actually see 2 points. Okay, magnification might just look like this. It makes it bigger. So if that's magnification without resolution. But resolution is not only the ability to make it larger, but to also see the detail, and that is the 2 points. So that you can see, yes, it's bigger, but it's also two distinctly different points. So a lot of times that has to do with the quality of the lens. So there's a lot of lenses that will magnify, but to have that resolution has to do with the quality of the lens. So a lot of times, you pay more money, you get better resolving lenses. It is also why the limit of resolution for light microscopy is actually 1,000 times. There's just so much resolution and magnification that you can do with the light microscope. Okay, and you just get maxed out at about 1,000 times. Any questions about that? Okay, so this is a little bit better explanation of magnification versus resolving power. So resolving power, as I said, is being able to identify two things as being separate. This is magnification without resolution. And if you have a better lenses, you can even resolve with better quality. Okay. Any questions about that? Okay. So one of the things that you would have done in this particular laboratory is that you would make your own slide of wet organisms, or live organisms, I should say, and it's called a wet mount. And this is where you would place a drop of the specimen on the center of the slide, and then you would use a cover slip by holding the cover slip at a 45-degree angle. And then you would draw the fluid on the edge of the cover slip so that it is continuous, just like you can see right there how it's continuous. And then you pretty much just drop the cover slip down over the top of the specimen. And that's how you make a wet mount. It's very easy. And then essentially you just put it on the microscope. So the type of organisms you would be viewing in this particular lab are protozoa. And these were, sort of the first unicellular organisms. So they're actually only a single cell, but you can see them, they're actually pretty large. Despite the fact that they're a single cell, you can actually see that they're a pretty big organism. And you can see this one is called paramecium, and it's a pretty big guy. The way it moves is through something called cilia. And cilia, as you probably know from your experience in the anatomy and physiology classes, cilia is just tiny hair-like um, structures that are usually anchored or rooted into the epithelium, into an epithelial cell on the apical surface. And pretty much that's the same that is true with the paramecium. And we can see in this sort of cartoon up here, we can see this paramecium, and we can see some of the, things that are most important about it. One thing that's important is the cilia, because that's how the actual organism moves around. It also has this thing called the gullet. That's where food will get actually in there. It too is actually ciliated, so it helps to scoop things into it so it can eat. But it has things like contractile vacuoles, and these are really important for organisms that are going to be living in aqueous environments like a paramecium, because they will help to allow the organism to move or to get at higher or lower depths in an aqueous environment. So what you can see, you can actually see these cilia on the surface, and remember that cilia can move on their own. They sort of have a beating motion. And if you were to see these organisms, and you would see that they do, they sort of beat on their own. And we're going to take a look at them live in a little bit. Unfortunately, we can't look at them under your own slides, but we can take a look using YouTube. Any questions about paramecium? So remember that they move by cilia. Okay, the next one is an interesting thing because it's a little bit more like a plant than an animal, because it has chloroplasts, and chloroplasts are typically associated with plants. They have a green pigment in them, and they absorb light, and they help to the organism to make energy. And so the way that these particular organisms move is that they have a single flagellum. So the single flagellum acts as a propeller, and it will enable the euglena to move through the actual aqueous environment. I don't know whether you can sometimes see here, the flagellum. You might be able to see the flagellum there. I think you can see the flagellum here. Okay, you can see the flagellum on these. Okay, so you can see them a little bit. But that's what actually allows them to move through their aqueous environment. And they're more like, plant-like, because they have, sort of that chloroplast. Okay. Any questions about euglena? The next one is an amoeba, and these things are very strange. They have something called pseudopodia, and it's their pseudopodia that allows them to move. Essentially, they have this amorphous shape. We sort of saw that the euglena and the paramecium, they had a very specific shape. And the paramecium had cilia to move, and the euglena had a flagellum. But the way the amoeba move is that they actually shoot the contents of their cell, so essentially their cell cytoplasm, into one of these things that are called pseudopodia. And the pseudopodia are really another name for false feet. And so that's what actually allows them to move, because they're going to shoot their cytoplasm into that sort of false foot. And then the rest of the actual cell is going to catch up with them. So that's pretty much how they work. Finally, the last one, which does not move, it just kind of has to go with the flow, if you will, is an anabina. And these are really interesting things because they look like just a whole bunch of little green beads, but they actually have, these represent cells, and they have sort of a granulation within the cells, and some of them are a little bit larger than others. So that's Anabina. They too are more plant-like because they have, green chloroplast-like pigments in them as well. Okay, so that's all for the PowerPoint. What I'm going to do is just show you some quick videos on how these organisms move. So, share again. So, okay, can you see this is amoeba? Okay, and I'm just going to play this a little bit. So you can see how the cytoplasm, these are the coolest things to look at. I have to say it's a shame not to get to see them, although sometimes they can be a pain to see in the lab. But you can see how their cytoplasm actually move into these extensions of the cell. And they will engulf things and eat them as well by doing that. Amoeba is really funny because, for years we kind of thought amoeba to be, uh, sort of, it's kind of delicate. But you can see this organism, which looks like a paramecium, is having either trying to eat the amoeba or vice versa. Um, but now we know that there are some forms of amoeba that are very dangerous. If they get into your nostrils, because of the way that they move, they can actually get into your brain and eat your brain. So people that use those neti pots, you want to make sure that the neti pot, the fluid that you use in neti pots, are as completely sterile, free of any life form, because if there's any amoeba in there, they can get into your brain and eat your brain, and then that's fatal. So very serious. But you can see how this happens. It's pretty interesting and wild looking. Okay, so next, that was the amoeba. This is the Euglena gracilis. This is the one that's green that has the flagellum. So this is how they move, by a single flagellum, and they're doing some crazy things there. Yeah, some of them have some funny different other shapes that they go into as well. And then finally, this is paramecium, and paramecium is pretty cool too. These are the ones that have the tiny cilia all around their edges, and you can see those cilia, they're just beating away as fast as they can, allowing this organism to move. Alrighty, so that's all I have for you today. Does anybody have any questions?
I have a quick question.
Sure.
Would you be able to post the quiz right after this class? We can do it today, or would
Yes,
you
yeah, I'm
post
going to
it
do it
tomorrow?
as soon. Takes me a little while because it's going to be the type of quiz that will actually be able to be graded, just like I won't have to interact with it, I hope. So I'll let it grade itself, and that way you'll have your, exams, your answers graded as soon as you've taken it.
Okay, so it will
Okay.
be up today.
Yes.
Okay, thank you.
Okay, you're welcome. Does anybody else have any questions? Alrighty then. Well, I thank you very much. I hope you have a great afternoon. And yes, I will post the quiz as soon as I possibly can.
Thank you.
Thank
Okay.
you.
Thank you.
Thanks, everyone. Have a good afternoon. Bye.